Welcome to the Bodhi Academy. My name is Shrutika and today let us continue with our chapter Photosynthesis in Higher Plants. So in our previous classes we have covered what is photosynthesis. We have seen what are how photosynthesis occurs in plant, and we have also seen that the structure of chloroplast and how in the chloroplast photosynthesis process occurs. So today what we are going to study is Calvin cycle. So what is Calvin cycle? So Calvin cycle can be described under three stages. So the Calvin cycle undergoes three stages, that is carboxylation, the first stage is carboxylation, then reduction and regeneration. So how these things happens in the Calvin cycle? These things happens in a cyclic form, that is first the carbon dioxide from the atmosphere combines with the water. So carbon dioxide plus water will form the carboxylation process, and from this what you will get is the 3-phosphoglycerate. This 3-phosphoglycerate will undergo reduction, and from ATP and NADPH it will form ADP plus NADP. Then after that it will undergo triose phosphate, and then that triose phosphate will form sucrose or starch, and then that triose phosphate will undergo regeneration to form from ATP it will form ADP. Then again that will undergo, after regeneration what is formed is ribulose 1,5 bisphosphate. Then this cycle continues, and again it will undergo carboxylation, reduction and regeneration. So this is how Calvin cycle takes place in plants. So carboxylation, so it is the first step, that is it is fixation of carbon dioxide into 3-phosphoglycerol glyceric acid, that is 3-PGA. Then carboxylation of RUBP, that is ribose bisphosphate, occurs in the presence of enzyme RUBP carboxylase, that is RUBS co, that is ribulose bisphosphate co, that is carboxylase, which result in the formation of two molecules of 3-PGA, that is 3-phosphoglyceric acid. Then it undergoes the second step, that is reduction. So reduction is a series of reaction that leads to formation of glucose. So two molecules of ATP and two molecules of ATP and two molecules of NADPH are required for reduction of one molecule of carbon dioxide. Then 6 turn of these cycles are required for removal of one molecule of glucose from the pathway. Then it undergoes regeneration. So regeneration is the generation of RUBP molecules for the continuation of the cycle. So in the regeneration phase what you get is RUBP molecule for the continuation of the cycle, so that the cyclic process continues, and this process requires one molecule of ATP. So this is how Calvin cycle takes place, through carboxylation, that is the first step, then it undergoes reduction and then the regeneration for continuation of the cycle. Then let us see what is C4 pathway. So this is a pathway which was found by H and Slay in 1965 and 1967. So mainly operational in plants growing in dry tropical regions like maize, sugarcane, sorghum. So C4 pathway is basically seen in maize, sugarcane, sorghum and plants which grow in dry tropical region. So in this pathway, first stable product is a 4 carbon compound which is called as oxaloacetic acid. So the first product in this cycle is oxaloacetic acid, so it is called as C4 pathway. Then the C4 plants have Kranz anatomy, that is vascular bundles are surrounded by bundle sheet cells arranged in grid-like manner. So characterized by large number of chloroplasts, thick wall impervious to gases and absence of intercellular spaces. So the anatomical part of this plant will show the large number of chloroplasts, thick walls to gases and absence of intercellular spaces. So the primary CO2 acceptor is a 3-carbon molecule phosphophenol pyruvate, okay, present in the mesophyll cells, and the enzyme involved is PEP carboxylase. Then OAA, that is formed in the mesophyll cell, forms 4-carbon compound like malic acid or aspartic acid, which is transported to the bundle sheath cell. Then in the bundle sheath cell, it is broken down into CO2 and a 3-carbon molecule. Okay, so in the bundle sheet it is broken down into CO2 and a 3-carbon molecule. Then the 3-carbon molecule is returned back to the mesophyll cells to form PEP, that is phosphoenol pyruvate. Then the CO2 molecule released in the bundle sheet cell enters the Calvin cycle, where enzyme RUBIS-CO is present that forms the sugar. So this can be explained using a diagram, that is the C4 pathway diagram. So over here we can see that the main processes that C4 pathway undergoes is the atmospheric carbon which is taken, it is first fixed, then transported, then recarboxylation takes place, after recarboxylation the C3 acid is transported, and then again regeneration takes place in the bundle sheath cell. So now let us see what is photorespiration. So photorespiration, it is a light dependent process of oxygenation of RUBP and release of carbon dioxide by photosynthetic organs of the plant. So photorespiration decreases the rate of photosynthesis when oxygen concentration is increased from 2 to 3% to 21%. So presence of light and high concentration of oxygen results in the binding of RUBIS-CO enzyme with oxygen to form PGA and phosphoglycerate. So RUBIS-CO enzyme will bind with oxygen to form PGA and phosphoglycerate. So this is the process which happens in the photorespiration in plants. So now let us see what are the factors affecting the rate of photosynthesis. So the first factor in this process is light. So as light intensity increases, the rate of photosynthesis also increases until light reaches the saturation point. So after reaching the saturation point, it is not applicable. So you require appropriate intensity and light period for the plants. So the photosynthetic rate will be optimum or will increase when the light intensity increases in plant. Second factor is carbon dioxide concentration. So with increase in concentration of the rate, so increase in carbon dioxide concentration shows increase in concentration of the rate of photosynthesis and increases till the compensation point. So it has a compensation point. Till that it will show increase in rate of photosynthesis. Then temperature. So it does not influence the rate of photosynthesis directly, but at a higher temperature enzyme activity is inhibited due to denaturation of enzyme, which will affect the dark reaction. So what is the dark reaction? Dark reaction is basically the respiration process. So temperature, if it is increased, the enzyme which are present in the plants are basically proteinaceous enzyme. So protein will denature at higher temperature. So the plant will not have enzyme which will be required for the processes of the plant. So of the plant, that is photosynthesis and respiration. So that is the reason why temperature is also an important factor in the process of photosynthesis. Then the fourth factor is water. Due to increase in the amount of water, rate of photosynthesis does not increase proportionally, as after saturation no more water is required during the photosynthesis. So basically water is not that effective factor in the process of photosynthesis, because increase in water level will not increase the photosynthesis process, because at the point of saturation no more water is required for the process of photosynthesis. So in this video what we have covered is what is Kelvin cycle, then we have seen what is CFO pathway, and we have also seen which are the various factors which affects the rate of photosynthesis, that is temperature, water concentration, carbon dioxide concentration and light. So thank you for watching today's video. Please don't forget to like, comment, share and subscribe to the Bodhi Academy.